At this stage of your immune knowledge, you must be aware of the important role that cytokines play in propagating the immune response. Without these soluble protein molecules and the network that they form, the immune system cannot function properly. Now the question that we are going to answer in this video is which cytokines are secreted by the T cells. As you may be aware that the T cells play a major role in the adaptive immune system. In fact, the cell-mediated arm of the acquired immunity is carried out by the T cells. T lymphocytes are a major source of cytokines. These cells bear antigen-specific receptors on their cell surface to allow the recognition of foreign pathogens. They can also recognize normal tissue during episodes of autoimmune diseases. There are two main subsets of T lymphocytes, distinguished by the presence of cell surface molecules known as CD4 and CD8. They are the cellular fighters that fight against invading pathogens. Let's go through the names and functions of these cytokines one by one. So, the first cytokine that we will talk about is interleukin-2. Once T cells are activated, they secrete interleukin or IL-2. IL-2 is a potent pro-inflammatory cytokine. T lymphocytes both secrete IL-2 and express receptors for it so that they can stimulate their own proliferation. This is known as the autocrine effect. Other functions of the interleukin-2 include the promotion of further growth and differentiation of activated T cells. Its effects include the proliferation of cytotoxic T lymphocytes, T helper cells, and natural killer cells, while enhancing the secretion of other cytokines, such as interferon gamma and TNF tumor necrosis factor alpha. Another cytokine produced by the T cells is interleukin-3 or the IL-3 regulates the growth and differentiation of hematopoietic progenitor cells and functionally activates mature neutrophils or macrophages and mast cells. IL-3 synergizes with other cytokines to stimulate the growth of immature progenitor cells of all lineages, and it is also recognized as the multi-lineage colony stimulating factor, CSF. Some other examples of the cytokines secreted by the activated T cells are: interleukin-15 stimulates differentiation of cells, just like interleukin-2. It also stimulates the B cells, leading to immunoglobulin synthesis and inducing the maturation of dendritic cells. Thus, boosting interleukin-15 activity could enhance innate and specific immunity and fight tumors. Lastly, we have interleukin-17, which is a pro-inflammatory cytokine. It plays a key role in linking T cell activation to neutrophil mobilization and activation. Earlier, I mentioned that the T lymphocytes are subdivided into CD8 and CD4 cells. T lymphocytes expressing CD4 are also known as helper T cells, while those that express the CD8 protein are called cytotoxic T cells. The CD8 cells mainly secrete cytotoxic molecules like perforin and granzyme B. They also produce antiviral cytokines such as interferon gamma and tumor necrosis factor alpha. The CD4 cells are regarded as being the most prolific cytokine producers. This subset can be further subdivided into Th1 and Th2. The cytokines they produce are thus known as Th1-type cytokines and Th2-type cytokines, respectively. First, let's look at the Th1-type cytokines. These cytokines tend to produce pro-inflammatory responses. They are responsible for killing intracellular parasites and for perpetuating autoimmune responses. Cytokine known as the interferon gamma is the main Th1 cytokine. The effect this cytokine has on other cells of immunity are: It stimulates the macrophages to kill phagocytosed pathogens. It also activates the natural killer cells so they can kill the virus-infected cells. IFN-gamma increases the expression of major histocompatibility complexes and antigen presentation by cells of the immune system. Lastly, interferon-gamma also inhibits the differentiation of Th2 cells. Excessive pro-inflammatory responses can lead to uncontrolled tissue damage, so there needs to be a mechanism to counteract this. Now if we talk about the Th2 type cytokines. This group includes interleukins 4, interleukins 5 and interleukins 13, which are associated with the promotion of IgE and eosinophilic responses in atopy. And also, interleukin 10 has more of an anti-inflammatory response. If we look at interleukin 4 a little closer, we can see that it has three main functions. The first function of IL-4 is that it induced the differentiation of T cells into Th2 cells, that is, the helper T cells. Interleukin 4 also promotes the growth of B cells, as well as enhances the class switching of IgE and IgG antibodies. These functions of the cytokines interleukin 4 can be remembered easily with help of a mnemonic. I am not too proud to beg for help. Focus on the coloured letters in this sentence. If you memorize this, you can link the functions of interleukin 4 with the coloured letter. For instance, here 2 is for Th2 cells to remember the first function of the cytokines, which is the differentiation of cells into Th2 cells. Next, we have the word beg. B is for the B cells growth, while E and G are associated with the class switching of antibodies IgE to IgG. For help is the indicator that the aforementioned functions are of interleukin 4, which is secreted by the helper cells. Another cytokine released by the Th2 helper cells is interleukin 5. This cytokine promotes the growth and differentiation of B cells, and also stimulates the growth and differentiation of eosinophils. It also enhances the class switching of Ig antibodies. Memorizing the functions of IL-5 is made easy with the help of this mnemonic. Eosinophilic bag, where each coloured and capitalized letter is associated with the function of the cytokine interleukin 5. The third Th2 type cytokine is interleukin 13. The effects of IL-13 on immune cells are similar to those of IL-4, as these cytokines share a common receptor subunit. Although IL-13 plays a predominant role in allergic inflammation by promoting bronchial hyperreactivity and overproduction of mucus, IL-13 also promotes isotype switching of B cells to produce IgE. Lastly, we have interleukin 10, which acts as an immunoregulatory molecule. The IL-10 cytokine decreases the expression of MHC class II and Th1 cytokines, effectively suppressing the immune response to some degree. It also has an inhibitory effect on both the macrophages and dendritic cells. Therefore, it has an anti-inflammatory effect and conveys signals of suppression to other cells. Interleukin 10 is a regulatory cytokine that is also secreted by the T regulatory cells. Research on the cytokine secreted by both Th1 and Th2 cells has concluded that, in excess, Th2 responses will counteract the Th1-mediated microbicidal action. The optimal scenario would therefore seem to be that humans should produce a well-balanced Th1 and Th2 response suited to the immune system. That is the answer to the question of which cytokines are secreted by T cells.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.